Hello, and welcome everyone to JDRF's December Research Information Volunteers Call. This is on disease-modifying therapies for T1D and is part 2 in our DMT topic series. I'd like to introduce today Joshua Veith, Director of Research for JDRF's Cures team, and Jay Tinklepaw, Scientist for JDRF's Cures team. Josh, over to you.
Thank you, Rachel. As Rachel said, my name is Joshua Veith, and I joined JDRF in September as Director of Research for Disease-Modifying Therapies on the Cures team. I'm very happy to be able to come and speak with you today about some of the exciting research we have going on in our disease-modifying therapy pipeline. And out of the expansive JDRF research priorities, today we're going to focus on disease-modifying therapies and the work being done in that field. Now, disease-modifying therapies are therapies that can change the course of a disease, and they're aimed at people at every stage of type 1 diabetes. From stage 1, where you have normal blood sugar and you're expressing autoantibodies, all the way to stage 4, where you have established type 1 diabetes. And the overall goal of our disease-modifying therapy program is to prevent, halt, or reverse the loss of beta cells. Then this therapy can be directed either towards the immune system, the beta cells, or both. Now, the focus of our disease-modifying therapy strategy is really twofold. One, it's to turn off the autoimmune attack against the insulin-producing beta cells, and I'll be speaking about that today. And Jay, my colleague, will be speaking about the second part, which is creating and sustaining beta cells in the body. And in both cases, our goal is to include treatments that both control the autoimmunity and preserve or regrow beta cells. And JDRF is accelerating the development of these therapies into clinical testing and eventually regulatory approval and access. So for the first part of this talk, we're going to focus in on turning off the autoimmune attack against the insulin-producing beta cells. And really a great place to start is with the immune system itself. And ultimately, the primary function of the immune system is to determine what is yourself and what is not yourself. And an inability for the immune system to properly determine self from non-self can lead to the development of autoimmune diseases such as type 1 diabetes. It's also important that therapeutic options that we develop create a balance. We want to maintain the ability of the immune system to surveil and recognize and eliminate threats, but we want to keep it from being so active that the immune system recognizes and attacks your own cells. If we look at the graphic, as you've seen before, of the stages of type 1 diabetes, the important thing to remember is that the immune events, the turning on of the immune system against your own cells, happens long before stage 1. This is where the autoantibodies that we use for screening come from, and it's why the screening that Frank spoke about last month is so important. So the recognition of self and the activation of the immune system initiates prior to stage 1 disease. So therefore, it's important not only to understand what events lead to this immune activation, but also develop therapies that can not only stop it, but can treat patients at every stage of disease. Now, if we focus in on the disease-modifying therapy development pipeline, last month, Frank discussed some of the clinical trials that JDRF is supporting to test candidates for disease-modifying therapies. And then Mariana spoke about some of the advocacy work that JDRF does to represent the interests of patients and families, and to ensure that the therapies that are in development can make their way into hospitals and into doctors' offices. The ultimate goal of this is better outcomes, to identify and develop a disease-modifying therapy that can prevent, halt, or reverse the onset of type 1 diabetes. For the purposes of my talk today, I'm going to talk about the beginning of the pipeline. I'm going to talk about the discovery research that is involved in identifying new targets and testing new ideas for therapeutics. And it's this development research that ultimately feeds the pipeline and ensures that JDRF continues to support studies and science that represent the next round of clinical trials and a potential cure for type 1 diabetes. Now, our strategy for turning off the immune attack against beta cells is threefold. First, we want to turn on the brakes of the immune system. We want to slow it down. Second, we want to interrupt the recognition of beta cells by the immune system. And finally, we want to turn down the inflammation and the negative effects that come along with immune activation. Now, when we talk about immune activation, we're talking really about recognition, and it's antigen recognition. And an antigen is really any particle that is recognized by and can activate the immune system. We generally think of these as potentially bacteria or viruses that invaded the body and need to be eliminated. It can also be toxins or proteins that need to be eliminated. But really, an antigen in its simplest form is anything that can be recognized by and activate the immune system. And that's the important thing to remember. Now, our immune system has cells that are able to take in these antigens and then present them on their surface to other cells in the immune system. These are what we refer to as antigen presenting cells, and you may have heard this term before. Some types of antigen presenting cells are dendritic cells and macrophages, and I'm sure you may have heard these in other talks. But for the purposes of our talk today, we're just going to call these generally antigen presenting cells. And they do exactly that. They flag an antigen and present it to an immune cell, another cell in the immune system that recognizes it and then can go and do a certain job. Now, when this antigen, for a number of reasons, comes from cells in your own body, it's called an autoantigen. It's an antigen your immune system is recognizing itself. So autoantigens are antigens from your own cells that are recognized by and activate the immune system. In type 1 diabetes, your immune system is recognizing autoantigens from beta cells, and the immune response to these beta cell antigens is what makes type 1 diabetes an autoimmune disease. And this is the basis for our research strategy in disease-modifying therapies. The immune system is very complex, and it's easy to get overwhelmed by the jargon and the processes that are described. So for the purposes of today's talk, I'm going to focus in on 3 cells in our immune system that recognize antigens presented by the antigen presenting cells. And these are B cells, T cells or T effector cells, and T regulatory cells, what we call Tregs. Let's focus in on just B cells for a moment. When a B cell recognizes an antigen that's being presented, it activates and produces antibodies. Now, these antibodies are small Y-shaped proteins that also identify and target and can recognize that antigen. And they go out and they tag this antigen for destruction by the immune system. They're almost a flag that says, hey, it's time to get rid of this. Now, in the case of autoimmunity, let's say that antibody recognizes an antigen on your beta cell. Because it's recognizing yourself, this is what's called an autoantibody. And you've probably heard this term before. We screen for autoantibodies at the earliest stages of type 1 diabetes. These autoantibodies can be pathogenic, meaning that they can mark your cells for destruction and lead to further immune activation and further progression of type 1 diabetes. Some of them are also diagnostic, whereby they don't necessarily activate the immune system further, but they're a sign that your immune system has already started to recognize your beta cells and started to mount an immune response against them. And some of the common diagnostic antibodies we talk about in type 1 diabetes are ICA, GAD65, IAA, or IA2A, and you may have seen these in the literature as you're reading. These are the autoantibodies we screen for in stage 1 and stage 2 of type 1 diabetes. Now, some work that JDRF is funding to halt this autoantibody production and the activation of B cells, some of it comes from Dr. Julie Ziekerman at UCSF. And she's investigating a new way to identify the B cells that recognize self-antigens from beta cells. Her focus is on a family of proteins called NR4A. And NR4A is expressed in B cells that are receiving a constant go signal or a constant activation signal. And this suggests that these are the B cells that are ultimately responding to the self-antigens, perhaps the antigens from your beta cells. So what Dr. Ziekerman is doing is she's developing tests that can find these NR4A positive B cells to identify them in patients. She's also using this ability to identify these cells to figure out which specific self-antigens that are activating these cells. And then with this information, to develop a therapeutic drug that can target only these NR4A positive cells, with the idea being that the self-reactive B cells, or the B cells that are recognizing your beta cell antigens, are depleted, leaving the other B cells in the immune system that are doing their job normally to continue working. Another set of research going on in the B cell field is coming from Dr. Todd Brusco at the University of Florida. Now, JDRF has a history of funding Dr. Brusco through postdoctoral fellowships, career development awards, and now through this ongoing research agreement. And this really highlights the importance of investing in young scientists and bringing them into type 1 diabetes research, because it allows us to develop the next leaders in the field. Now, Dr. Brusco's idea is that the B cells that produce autoantibodies are an early target for halting or reversing the onset of type 1 diabetes. And he works on another type of cell in the immune system called a T follicular regulatory cell. And really all you need to know about a T follicular regulatory cell is that it's able to stop B cells from being activated. So using cutting edge gene engineering, he's using CRISPR-Cas9 to create T follicular regulatory cells that not only have a receptor that can recognize the self antigens from beta cells, but also have another receptor that traffics or causes these cells to migrate to areas of the body where the B cells are being activated. And the idea here is that when you have the presentation of a self antigen to a B cell, these cells will be present to inhibit that activation and stop the B cells from being activated and producing autoantibodies. And he's currently doing tests to see if these engineered cells can halt type 1 diabetes in an animal model in preparation for further clinical testing. The next type of immune cell we're going to focus on are the T effector cells, and Frank spoke on these briefly last month as well. These are the cells that, once activated, go out and seek and destroy their target, the antigen they recognize. Now, when a T effector cell recognizes antigen presented by an APC, it activates and proliferates. So by activate, it means it turns on. And by proliferate, it means that it creates multiple copies of itself. And each of these copies recognizes the same antigen, and they go out to try to find this antigen and destroy the cells associated with it. Now, in the case of type 1 diabetes, if this antigen happens to be on a beta cell and one of these T effector cells recognizes it, they activate and they produce a chemical. They produce a chemical we call cytokines, and these cytokines have a number of results. First, they cause inflammation in the surrounding tissue. This is where we sometimes see pancreatitis or tissue inflammation in association with type 1 diabetes. The next thing they do is they actually destroy the cell that's recognized, and the destruction of the cell causes the release of more autoantigens and further activation of the immune system. So it's a process that continually ramps up itself. And some work that JDRF is funding in this area comes from Dr. Angela Lombardi at Einstein College of Medicine. And she's looking to block the expansion of these autoreactive T cells. Now, what Dr. Lombardi has done is she's developed a small molecule called D peptides. And these are basically synthetic versions of the antigen that's recognized by your self-reactive T cells, connected to a protein that mimics the presentation from the antigen presenting cells themselves. But it's been modified in a way that when this small molecule binds to your effector T cells, it doesn't allow them to be activated. Now, this has an added bonus in that because these D peptides are bound to your T effector cells, it also stops the antigen presenting cells from presenting the antigens from your beta cells themselves, meaning that the goal here is that the T effector cells are never activated and aren't able to go out and attack your beta cells further. Currently, Dr. Lombardi is testing the ability of these D peptides to stop the activation of the self-reactive T cells. She's investigating whether you can use the D peptides to halt and or reverse type 1 diabetes in an animal model. And she's also doing work in T cells isolated from type 1 diabetes patients to validate this as a therapy for clinical trials. Now, one thing I'd like to also say about Dr. Lombardi is this work was funded through a one-year innovative award. And it's nearing completion and has been very successful. And this work is actually being expanded into a fully funded NIH study. And she's also looking to initiate a phase 1 clinical trial for the use of D peptides. And this is a great example of how important the innovative grant program is here at JDRF. It really enhances our ability to facilitate the transition of great novel ideas and discovery research into the next round of clinical trials and therapies. The final cell we're going to talk about today are the T regulatory cells, or Treg cells. And the best way to think about Tregs is that these are the good guys of the immune system. These are the cells that once activated actually go out and they slow down the immune process. They shut down the immune system so it doesn't become too active. So just like other T cells, when they recognize antigen that's presented, they activate and proliferate. So they turn on and they create copies of themselves that recognize the same antigen. But what differentiates them from other T cells, such as T effector cells, is that when a Treg recognizes, let's say, an antigen on a beta cell, instead of producing those inflammatory cytokines or chemicals that cause destruction, they produce chemical messengers that actually calm down and soothe the inflammation. And if there are effector T cells in the area that are attacking beta cells, these chemicals actually put these cells to sleep. They turn off the immune response against the beta cells. So Tregs are a really important focus of therapy for our disease modifying approach. And this is a process called immune regulation. And this is really the goal. We want to be able to highlight the body's ability to regulate its own immune system and have a targeted way to shut down the self-reactive immune response. JDRF is currently funding work by Dr. Jordan Green at Johns Hopkins. And Dr. Green is actually not an immunologist, he's a biomedical engineer, and he has a very unique approach to try to tolerize or turn off the immune system using Tregs. Dr. Green has developed what he calls artificial tolerizing APCs. Now, we talked about those antigen presenting cells. Dr. Green is creating a synthetic version of these out of a number of materials, and he's calling these TOL APCs, or artificial tolerizing APCs. And these synthetic molecules have the beta cell antigen that's recognized by T cells on their surface, as well as another molecule that can strongly activate T regulatory cells. So they turn on the Tregs. Now, this molecule inhibits the activation of T effector cells. So in effect, when you add these tolerizing APCs, they flip the switch for the immune system into more of a protective than an effector or an attacking state. And some of the ongoing work by Dr. Green includes testing a range of these synthetic molecules for size, shape, and also the materials they're made of for their ability to interact with T cells. He's also testing their use in an animal model to allow for or to demonstrate their effectiveness and whether they have therapeutic potential for clinical development. Finally, JDRF funds research around the world. And a great example of this is the work being done by Paolo Monti at San Raffaele in Milan. Now, JDRF has funded Dr. Monti through postdoctoral fellowships and through career development awards, and all of which were very successful and resulted in the ongoing work in his lab that JDRF is also funding. And he's focused on identifying and developing Tregs that are optimal for immune suppression. Now, past work in Dr. Monti's lab has shown that transplantation or putting Tregs into T1D patients is both feasible and safe. You can place these cells into patients and they do shut down the immune response. However, initial experiments haven't been overly successful because it shows that these cells, while they initially have an effect on the immune system, they aren't long-lived after transfer. They don't last very long in the recipient. So it's not been an effective approach to have lasting immune tolerance. Now, Dr. Monti's ongoing work has taken T cells or T regulatory cells from type 1 diabetes patients, and he's developed a new method in his laboratory for expanding and activating these cells in vitro, meaning outside the body or basically in a test tube or plate. And this new method that Dr. Monti has developed gives these Tregs stem cell-like properties. And what does this mean? So when he takes these Tregs and puts them back into his model system, he shows that they self-renew. So they continually create new copies of themselves so they stay around. They also have very high immune suppression potential, meaning that they work even better than normal Tregs. And finally, they persist after transfer, meaning that this is potentially going to get past our issue with Tregs of not being long-lived, and that this could be a lasting therapy for type 1 diabetes patients. And Dr. Monti is currently testing this novel expansion protocol in a preclinical model and validating it as a potential cellular therapy in clinical trials. And I hope that the take-home message for you from my part of the talk is that this discovery research that JDRF is funding leads to the future therapies, the future clinical trials. And even if you don't understand all of the immunology and complex science that underlies the JDRF research strategy in disease-modifying therapies, the takeaway message is that we're funding discovery research in the US and around the world to develop a wide variety of approaches. And as you can see in this slide, we're investigating a number of approaches in multiple parts of the immune system to prevent or stop the immune attack on beta cells and eventually develop a cure for type 1 diabetes. And now I'm going to turn the talk over to my colleague, Jay Tinkelpaul, who's going to discuss targeted therapies for creating and sustaining beta cells.
Great, and thank you very much, Josh. Hello, everyone. My name is Jay Tinkelpaw. I'm a scientist at JDRF, and today I will be discussing JDRF's ongoing work to develop and advance disease-modifying therapies that are focused on the beta cells, and really looking to answer the question of how do we create and sustain them. Specifically, JDRF is supporting projects that are centered on finding new proliferative targets to encourage beta cells to replicate or proliferate. And we are also supporting projects that are implementing new targeted drug delivery-based approaches. So promising drugs that help the beta cell either proliferate or regenerate are delivered only to the beta cell and not to other cells throughout the body. To address this challenge of how to create and sustain beta cells, JDRF has really broken this down into three broad areas: identifying new ways to improve beta cell survival and function, finding ways to improve the function of beta cells that have survived the immune attack and encourage them to replicate or proliferate, and finally, finding ways to convert other cells into insulin-producing beta-like cells. Now, Frank and Josh have done an excellent job of highlighting many of the disease-modifying therapies that address the immune component of T1D. I will be focusing on ways we are trying to protect the beta cell and encourage more beta cells to grow. So as you know, in T1D, we classify the population into 4 distinct stages. Currently, most disease-modifying therapies are targeted at the stage 3 population. That's due to a number of reasons. One of them being that many individuals in stage 3 still have a measurable number of beta cells remaining. Thus, there's a reasonable number of targets to deliver a drug to. However, while the projects I will be discussing are starting in the stage 3 population, the goal is these beta cell focused strategies will help those with all stages of T1D, including those with established T1D in stage 4, who may only have a very small number of beta cells left, and those in stages 1 and 2, keep them from progressing to insulin dependence. So as I mentioned, the first approach is centered on finding new ways to improve the survival and function of beta cells in response to stress and the attacking immune system. Now, not every beta cell dies solely due to the immune system. And when the beta cells are stressed, they can begin to lose function and ultimately die during T1D. So previous JDRF research has identified several different pathways and drugs that can keep beta cells alive and functional by making them more resilient to the immune attack. An example of a drug that we think does this is verapamil. So verapamil is a drug primarily used to treat high blood pressure, but in mice with diabetes, it has been shown to reduce beta cell death, which you can see in the figure. Diabetic mice that were not given verapamil had much more beta cell death and thus significantly less insulin production than those that received the drug. Currently, JDRF is supporting verapamil's investigation in clinical trials to see if that same protective impact is seen in humans. The second approach I highlighted is focused on finding ways to get the beta cells that have survived the immune system attack and the associated stress to proliferate or multiply. The goal of this would be to replace the beta cells that were lost by encouraging the survivors to multiply and restore their overall number, as seen in the cartoon below. There have been several exciting discoveries in this area, and we now know how to make beta cells proliferate in vivo, but unfortunately not with any tissue specificity. And this is a huge problem that JDRF is trying to overcome, because proliferating cells in the wrong parts of the body could create some major safety concerns. This is a major objective of the targeted drug delivery projects that I will discuss later. And the third approach we are taking involves the conversion of one type of cell into something that much more closely resembles a beta cell. So as you may or may not know, as T1D and T2D, type 2 diabetes, progress, other cells in the pancreas try and change into the beta cell to help out and supply insulin to the body. Unfortunately, not enough cells are able to change from one type to another to make a difference, unless we still need insulin to manage type 1. We are studying ways to use gene therapy to deliver signals to other cells, both inside the pancreas and out, to force them to become beta cells. However, as you might expect, and similar to the proliferative approach, we will need to be very careful, and thus we will need highly controlled delivery methods and on-off switches to ensure that these strategies are implemented safely. So while there are definitely some very exciting tactics in progress in this area, there are still significant hurdles remaining. Mainly, can we induce proliferation in beta cells and or reprogram other cells safely without having dangerous off-target effects? And in order to address these problems, we're coming at them from a number of different ways. First, we're continuing to work on identifying new proliferative targets and strategies, and I will be discussing some early and ongoing work in that area through a partnership between JDRF and Boehringer Ingelheim. And second, in order to minimize the risks associated with inducing beta cell proliferation and or cell reprogramming, we are pursuing a number of projects in targeted drug delivery. A few of those I will also discuss today. So through a partnership between Boehringer Ingelheim and JDRF, there are 3 research projects really focused on finding new ways to proliferate and regenerate the beta cell. And just to reiterate, by proliferate, I mean, we want the existing beta cells to multiply or replicate. We want 1 to turn into 2 and then 4 and so on. Whereas regenerate, we want a stressed and potentially unhealthy beta cell to return to a healthy functional state. So the first project is with Dr. Bruce Verschere at the University of British Columbia, and it's based on the observation that certain immune cells can have flexible characteristics, with some being very pro-inflammatory and thus harmful to beta cells, and others being more beneficial and pro-regenerative. Dr. Verschere is working to identify the characteristics of these immune cells and potentially what they secrete that can help increase beta cell mass and hopefully find ways that these factors can enhance survival and the function of surviving beta cells. The second project is with Dr. Mark Housing at University of California, Davis, and is built on the observation that human beta cells have several different functional states. And by this, I mean that some beta cells are mature and are producing insulin, and others are at different stages in their development. They haven't quite reached that point. So Dr. Housing's project is really focused on identifying these different cell populations in the human pancreas and determining which of them can produce new beta cells, which of these populations might be the best for potential regenerative therapies. The hope is that these virgin beta cells, as he calls them, will be a promising target for the targeted drug delivery approaches that we are also pursuing. The third project is with Dr. Gregory Koo at UC San Francisco. And this is based on the well-documented observation that many studies to identify new pathways controlling human beta cell proliferation fail to translate from mouse models into humans. So Dr. Koo is taking a different approach to this problem and is attempting to use gene therapy to find new factors that trigger beta cell proliferation. The ultimate goal of this being to find new ways to both identify and validate new targets for human beta cell proliferation. Now, in addition to finding new ways to help beta cells replicate and be healthier overall, we're also working to develop ways to safely deliver drugs to the beta cell. And we are doing this via an exploration and implementation of targeted drug delivery systems. So targeted systems are designed so that any drug is delivered only to its site of action. For example, you wouldn't want a plane that you expect to drop you off in the sunny tropics to let you out in the Arctic Circle. Well, the same goes for drug treatment. Or as I've shown on the right here, we want to ensure that the drug is only going to the beta cell, the blue cell in this example, and not to any of the others throughout the body. The promise of this approach is that it mitigates the safety risk. Everything becomes safer, and it decreases the amount of drug that we would actually need for any treatment because it's just a more efficient approach. And so just like there are any number of ways you or I could get from A to B, there are a number of different drug delivery systems being looked at by JDRF, including using beta cell specific proteins as a form of drug shuttle, nanoparticles loaded up with regenerative drugs or compounds targeted specifically and only to the islets, and even viruses that will selectively reprogram certain cell populations and only those populations into beta-like cells. And today I'll be talking about 3 such projects in this targeted area. So the first project I'll be discussing is a partnership between Dr. Micah Sander at UC San Diego and Ionis Pharmaceuticals. So Ionis has developed a peptide-based technology that is able to deliver therapies directly to a human beta cell. Alongside this, Dr. Sander has developed with JDRF support a compound called an antisense oligonucleotide that can help make human beta cells proliferate in vivo. Naturally, these two things should go together. So JDRF has helped to create a partnership between Ionis and Dr. Sander to deliver these proliferative agents to the beta cell, and they are currently being tested by Dr. Sander in some mouse models of T1D. Now, if this project is successful, Ionis would be the first company to have a targeted drug system that can make beta cells proliferate. And this would be a very promising step on the path to a cure. The second project I'll discuss is with Dr. Paolo Serafini at the University of Miami. Now, Paolo's research is focused on using nucleic acid aptamers made of either DNA or RNA to deliver drugs specifically to the beta cells. So these aptamers, these short sequences of DNA or RNA, are very special due to the fact that they can fold into a shape that will fit only a very specific target. Think of them as the only key to a lock. This folding pattern allows the aptamer to bind tightly to its target so we can deliver a drug with very high specificity. If you look at the cartoon on the bottom of the slide, you can see a rough interpretation of this folding behavior on the right, and the remainder of this platform that Paolo is developing can be loaded up with a drug. So what Paolo's lab is currently working on are two of these platforms, both targeted to the beta cell, but designed to deliver different therapies. One is designed to improve beta cell function and potentially encourage some proliferation, where the other one is designed to help alleviate the immune attack on the surviving beta cells. His lab is currently testing these to determine how effectively they deliver the target and modify the cell. The final thing I will discuss today is a company from the T1D fund called CodeBio. Now, CodeBio is using scaffolds made from slightly modified DNA to serve as a customizable drug delivery platform. So as you can see in the cartoon, there's a DNA scaffold framework, the double-stranded helixes, that can be then customized with different antibodies, different drugs, different imaging agents. The list goes on and on. And it's really this flexible nature of the platform that makes CodeBio's scaffold so exciting, because in addition to all of the different therapies that can be attached, they can be modified with almost any type of targeting agent that confers specificity to the beta cell. So that could be a peptide, an antibody, small molecule, numerous different options. So to summarize, currently targeted drug delivery approaches are predominantly in the proof of concept stage in T1D, but we at JDRF and across the field really are working to keep them moving forward. Right now, JDRF has small peptide, aptamer, viral, and non-viral projects in the development pipeline, but we expect to have many more in the near future. Some of these projects include nanoparticle delivery platforms, additional viral gene delivery methods, antibody drug conjugates, and so on. And we haven't ignored the immune system in this targeted drug delivery space as well. We are looking at how these targeted delivery strategies can be applied to the immune system and how the lessons being learned around targeting beta cells can also be applied to immune cells as well. And looking forward, the future of disease-modifying therapies is very promising. We will continue to identify discovery research that focuses on new and expanding areas of immune intervention, work to develop new combination therapies in type 1 diabetes that address both immune dysfunction and beta cell function, find and expand the number of targeted drug delivery approaches we have for both the immune system and the beta cell, and really continue to drive discovery research into the clinical setting with the goal of developing a cure for T1D. Thank you so much for your time, and we look forward to hearing from you on the Q&A call.